is does this medication that's being studied, does it affect survival? That's often what we really want to know. Do people live longer with this medication? But that's not really very easy to study. It would take a very long time and a lot of people to do something like that. So we often use what is called a surrogate outcome. And that is essentially a shorter term outcome where it is a proxy for survival. And so most commonly in pulmonary fibrosis trials, it's the forced vital capacity, or the FVC, which is part of the PFT. And it's actually something you can see over on the right in the screen, how we measure pulmonary function. When you blow into the machine, one of the values that you're getting is that forced vital capacity, and it's very predictive in pulmonary fibrosis. Other things we do look at include the 6-minute walk distance and extent of fibrosis on a CAT scan. But there are other important outcomes, and these are starting to become more and more important in trials as outcomes that are being monitored, including quality of life, how much oxygen people are using, how well they can do their everyday activities. These are incredibly important to pulmonary fibrosis patients and are being looked at as well. So who can participate in a study, and why is it that there are inclusion and exclusion criteria? Well, it's incredibly important that you get uniform groups of patients so the comparison is valid. It helps produce reliable results and make sure that the actual question you're asking with that study is answered, because there's no point in doing a trial if you're not actually going to get an answer from the trial itself. And it also helps to ensure safety. You don't want somebody in the trial who's taking a medication, for example, that interacts with the study medication. So there are reasons for these different things. And also if they have some medical illness that would make it unsafe for them to participate. So in IPF in particular, one of the main things is, do you actually have IPF or some other form of pulmonary fibrosis? And we'll talk about that a little bit more. Other pieces of information might include your age, pulmonary function data, what does the CAT scan look like, and are you taking other medications for your pulmonary fibrosis? So what are the challenges with this? Well, sometimes it's hard because somebody might want to participate and they don't qualify for a variety of reasons, even though they do have the disease of interest. Also, sometimes people with other forms of disease want to participate, but they can't because it's limited to very specific forms of pulmonary fibrosis. And sometimes a biopsy is required, and that may be something that a person didn't have or couldn't have. And what this means in the future is that in that trial, it may not apply necessarily to every single person with the disease, because we only know information after that trial from what was specifically studied, and sometimes we have to extrapolate later. That being said, there are a lot of reasons, and you actually, in your questionnaire, you really addressed some of these things really appropriately. You have access to a novel treatment before it's available generally. It's possible that this new treatment may be more beneficial than the standard of care. We don't know that, and which is why the trial is being done, but it's possible. You get close monitoring of your condition, and you can help others with the disease by contributing to research. And I think that that's actually one of the biggest things to really think about. There are always risks because there may be side effects that either are known or unknown about the drug. It's possible that the treatment won't work and that you'll have spent the time in a trial and we won't know that it works at the end. It's possible that the treatment will end up being worse than what we know about the care for the thing, and you're spending time and energy. And I think one of the last things here is people worry a lot about being exposed to illness. So I think it's important for us to actually spend some time talking about COVID, because this is the thing that people are very worried about right now. Can I go out and go participate in a trial? And so I think it's important to talk about safety and what's being done to protect patients as they're doing this. So first, clinics. So how do clinics manage this? Well, in our experience, there are a lot of questionnaires that happen before you come. So nobody with symptoms of infection is allowed to come in. There's usually separate facilities for anybody with sickness actively. Everybody's still wearing masks. There are the mask mandates that may be going by the wayside around the country are not going by the wayside in medical facilities, because ultimately the goal is to protect the most vulnerable throughout the facility. And so everybody is wearing masks. And there is so much hand sanitizing. If people could just mute their microphones, that would be great. There's a lot of hand sanitizing and cleaning that's going on at all times. And personnel there are very highly likely to be vaccinated. And the more vaccinated people around you, the safer things are. There's also the issue of what's going on in your community. So it depends on in your community what the numbers are looking like. And right now, in a lot of places, those numbers are really going down, which is amazing. And the less there is community spread, the lower your risk of infection in any location. And the question then is, what can you do? Well, obviously, I think it's very important for people to get the vaccine if they haven't gotten it already, to wear your mask, and to talk to your doctor about whatever concerns you have about these kinds of safety issues. I'll just show this. This is our new facility at Yale, the Winchester Center for Lung Diseases. And we're taking safety very seriously. We have negative pressures in the rooms. I can barely sit down at times because people are constantly coming and cleaning around me. We try to keep this facility incredibly clean and safe. Even our old facility also has negative pressure rooms. These are things that are incredibly important in protecting patients. But even in centers that don't have negative pressure, the protocols are in place for cleaning, for mask wearing. People are really considering patient safety, and there have not been outbreaks in these settings. So I wanted to spend a little bit of time in discussing how you find a trial. There are a lot of different ways. I've listed a few here. There's the Pulmonary Fibrosis Foundation, and clinicaltrials.gov is an incredibly important place. Your ILD specialist. If you live close to us, I've listed the website here. There's also Trials Today, which is also an incredibly organized way to search for a trial. People may not be savvy with how to find these things, so I just wanted to take a few minutes to just walk you through how you might do this on clinicaltrials.gov. So this is just to give you an example. So for example, if you go to the website, you can put in your diagnosis, a condition or disease, and search based on that, and then look over to the left. There's a way to apply filters. So you could, for example, put in studies that are recruiting, certain age criteria, other things to help you narrow down what you're looking for. You want to make sure that when you search, you're actually looking for the correct diagnosis or condition as to what you have. And actually, importantly, you can look by location. So there's actually a map feature here. So you can put in what you're looking for and then narrow it down by location. And then this is just to give you a sense for, if you click on Connecticut, you'll find this study that we're running. And you can scroll down in any given study and you'll see information, contact numbers. You can see the inclusion and exclusion criteria to see if your lung function fits the criteria, if the medications you're taking fit. So I hope that that's helpful in going through that. So I'm just going to switch gears a little bit and talk very briefly about IPF, idiopathic pulmonary fibrosis, and a little bit about trials in IPF. So what is IPF? Well, first and foremost, it's a very specific form of interstitial lung disease. I think it gets very confusing because of all the abbreviations that happen. So there's ILD, which is interstitial lung disease, and there's IPF, which is idiopathic pulmonary fibrosis. And IPF is a very specific form of ILD. ILD is the big umbrella category, and IPF is a specific disease. It tends to affect older adults. People tend to have progressive shortness of breath, cough, and oxygen dependence. So how do you define IPF? Well, number one, it's idiopathic. And what idiopathic means is that you have excluded known causes for the lung disease. So for example, there's connective tissue disease, things like rheumatoid arthritis, scleroderma, Sjogren's, for example. If you have connective tissue disease and lung disease, you by definition, no longer have IPF because there's a cause known for the interstitial lung disease. Similarly, there are other kinds of exposures, environmental exposures such as birds or mold, and medications that can cause lung disease. So for IPF, you need to have excluded all those things. And then on the CAT scan, you need something called a UIP pattern, or usual interstitial pneumonia. And if you do a biopsy, there should also be a UIP pattern. So what do I mean by UIP? This is just an example of what we're looking for on a CAT scan. So what you can see is on the left there, what we typically might see on a CAT scan, which is that there is scar tissue, particularly around the edges of the lungs, there are kind of stacks of cysts of scar tissue, and that's something called honeycombing. In the UIP pattern, it tends to involve the edges of the lung, the lower parts of the lung more than the upper parts, and it tends not to have a lot of inflammation. It tends to mostly have just scar tissue like this. And then if a biopsy is done, which is not the case for all patients, you'll see scattered areas of fibrosis. So what you see in this right here is areas of normal tissue. And where the tissue is normal, you see very thin alveoli, which is where the gas gets exchanged in the lung, the oxygen gets across. And the areas that are marked with an F are something called fibroblastic foci, and those are areas of focal scarring. And you can imagine how difficult it is for oxygen to get across in those areas of the lung. And this is very typical of what we see in a UIP pattern. So I just want to show you some examples of other forms of interstitial lung disease to show you how different things might look. And this is where your ILD specialist will take a look at the CAT scan and help you understand different forms of ILD that you might experience. So for example, there's something called NSIP, or non-specific interstitial pneumonia, which is most common in autoimmune or connective tissue diseases. So this is a picture of a patient's CAT scan who has scleroderma. And what you can see is there's kind of haziness in the lung fields, is graying, and that the scarring is more towards the center parts of the lung rather than at the edges of the lung. And so it's very different from what I showed you with that UIP pattern. And similarly over here, this is what it looks like when there's drug toxicity, where certain drugs can affect the lung and cause inflammation and scarring. This is something called hypersensitivity pneumonitis, where you can also see that there's honeycombing for this patient. You can see some cysts in the lung here, but this is the upper part of the lung. And these are the lower parts down here on the lower fields on the screen. And you can see that actually the scarring affects the upper parts of the lung more than the lower, which is the opposite of what we were talking about with the UIP pattern, which tends to affect the lower parts more than the upper. And so this is the kind of scarring that you can get over time from exposures, such as from birds in this particular case. So IPF itself, it may feel lonely. It may feel that it's a rare disease, but it's not. It actually is more common than certain forms of cancer that get more attention. And so that's why it's so important that support groups like this exist, because it is actually an incredibly common disease. And I think the more you look, the more you find patients who have something like this. It is a disease of aging. It tends to be seen more often in people who are older. You can see that in the different age categories listed here, it's incredibly uncommon in people under 50 years old. And as you go up from age 60 and over, you see it becomes much more common. It does lead to early mortality, meaning that people do die of this disease, which is why it's so important that we find medications not only to slow it down, but to stop it in its tracks and to really find a cure for this disease. So then that brings us to clinical trials for pulmonary fibrosis for IPF. So I just want to go through, there's a few lessons that we have learned over time in studying IPF in trials. And the first one is that trials are needed, and they need to be big trials. This was the first situation where this was really brought to the forefront of people's thinking. So in the late 90s, there was a drug called gamma interferon, and a very small study suggested that patients, and this was open label, meaning that everybody got the drug, so there was no placebo comparison group. And it looked suggestive that people had improved lung function and oxygenation, and people got very excited. And so it was being used in a widespread manner, and it was not a fun drug to take. It led to horrible flu-like symptoms, and people felt terrible. Eventually, a drug trial was actually done as a randomized placebo-controlled trial, and it turned out there was actually no benefit. So those patients who had been taking that drug and suffering all those side effects, it really had not been worthwhile. And the way we knew that was because of an actual large drug-controlled, placebo-controlled trial. So important. So lesson number one then is it's necessary to do the trials. Lesson number two is that unfortunately not every trial gives a positive result. Some trials will be done and it will show no effect of the drug. It does not mean it was not worth doing those trials, but not every trial succeeds, even with the best of intentions. And this is just to give you a sense that along the way, there have been a number of different trials and not all of them have shown a positive effect. But it was necessary to do those. However, sometimes a negative trial, and this is lesson number 3, sometimes a negative trial is actually a good thing. And so I want to give you some examples of why it's so important sometimes to have a negative trial. So for example, in the past, it was very common for patients with IPF to be treated with immunosuppressive medications. And so I mean prednisone and azathioprine in particular. And part of why that probably was before the current classification system for IPF, people were grouped together more. And so there were different kinds of diseases that were grouped together. And there were some patients in there that were grouped that probably did respond, because there are some forms of ILD that do respond to these medications. But after that, once IPF tended to be separated out from the other forms of lung disease, it was not clear that this was safe. And finally, a study was done. And what it actually showed was that patients died more, had more serious adverse events, and did poorly with this when they had IPF. And so we now know that for most patients with IPF, immunosuppression is not a good thing. I do want to put the caveat out there that there may be people here who are on immunosuppressive medications. And there are some reasons why patients with ILD are on immunosuppression. So for example, it may be that you don't have IPF and some other disease is being treated. Or sometimes after what we call an acute exacerbation of IPF, people might end up on immunosuppressive medications. So I don't want people to suddenly stop taking their medications. Obviously, talk to your doctor about this. But I do want to highlight this trial as a reason that we don't give it to regular IPF with moderate disease. Similarly, these are recent studies that came out. There had been a lot of interest in giving people regular antibiotics as a way to try to reduce exacerbation and treat pulmonary fibrosis. And ultimately, 2 negative trials have shown that that's not really what we should be doing, that regular antibiotic use should not be given and is not helpful. Lesson number 4, I think, is one of the biggest ones here, is that persistence and participation does pay off. This is from the New England Journal from 2014. And these 2 trials were published on the same day in the same edition of the journal. And what you can see here is the trial looking at pirfenidone and that looking at nintedanib. And what you see in the graphs of each one is that when you look at the drug compared with the placebo, the people who were receiving the drug, their lung function went down less. And that was incredibly exciting news. This was the first time that we had 2 drugs that were successful in slowing down progression of disease. And shortly thereafter, the FDA approved these drugs for use. And they have been incredibly important in slowing disease for many patients with pulmonary fibrosis. They're also showing use in other forms of pulmonary fibrosis as well. So this is the InBuild study. And what this shows is that for progressive fibrotic lung disease, not IPF, so other forms of progressive fibrosis, nintedanib also seems to slow the progression of lung fibrosis. Pirfenidone has also been looked at for progressive fibrosis, and we think it probably works. Unfortunately, these 2 studies were marred by difficulties with their actual setup. So for example, the first trial had to be closed because they didn't recruit enough patients, which is a major issue. And one of the reasons we're encouraging participation to try to get answers to these questions. And the second was they had poor outcomes. They tried to use a home spirometer. It was not reliable enough. And so we don't have definitive data about pirfenidone, but we think it may be effective based on the data that was available from these trials. But because the trials, number 1, didn't recruit well enough, we don't have definitive data, and pirfenidone is not approved by the FDA for progressive fibrosis. And then lastly, scleroderma, which is a different form of disease, Nintedanib also seems to slow progression of one's situation. You can turn off your microphone, please. Thanks. So that's some information about what we know to date about pulmonary fibrosis. And now I'm going to turn it over to Dr. Kaminsky, because this is one of the really key lessons that we've got to, which is that what we know now and what it comes in the future will lead to more hope for all of you. So I'm going to stop sharing my screen and turn it over to Dr. Kaminsky at this point.
Can you see my screen? And
this is
you see the slides progressing?
No, it's not coming through properly. No. Go try again, stop share. Oh, there you go.
Can you see it?
Yes.
Good. So again, thank you all. And we would like to have time to answer your questions. In the next, actually, probably 25 minutes, I'll speak about this important lesson that we've learned. Much of the research that was done in human pulmonary fibrosis, and actually a lot of the advance, was focused on thought that we had about what's happening in the human lung, based mostly on animal models of disease. And what we think is that the road to cure IPF depends on is actually on understanding pulmonary fibrosis in the human lung. And sort of the image you see on the right is a rendition of the different mechanisms. And I'll sort of revisit them in some simpler ways. The one and first things I want to tell you all is a very important thing, which is IPF research and pulmonary fibrosis research has exploded in the recent years. What you see here is basically numbers of publications in the general scientific literature from 2002. This is the baseline. And the sort of the dashed line is the average for all of the papers. Oh, and the yellow is coronary heart disease and brown is cancer. So these are things you know, there's a lot of research and a lot of money, way more than in pulmonary fibrosis. But the red line is the number, the increase in the numbers of research papers in pulmonary fibrosis. And this is important because usually when there is more research, you usually get to an increase in therapies. And this dramatic increase in good research and a lot of discoveries is really important. And if you look at it, there's several drivers to this process. One is in 2011, and I'll speak about it quickly, 2011, 12, and 13, we made sort of key discoveries about the genetics of pulmonary fibrosis, which sort of drove the field forward because suddenly we have started to understand what is the disease in humans. Second is in 2013, we had the approval of 2 drugs. And usually when 2 drugs work, the first 2 drugs are never perfect, but they drive the process. So it's a little bit like I started my career at the heights of the AIDS epidemics. Patients were dying everywhere. And then I remember in 1997 in the San Francisco Chronicle, the headline said, they thought they were going to die. Now they need to find jobs. And this was the effect of all the new drugs and AIDS, and it take a few years. So usually the first 2 drugs are not perfect. They slow down disease. They help, but they have side effects. But then the next drugs do well. So, and definitely this approval of drugs brought in a lot of money. And then the last is sort of something I'm hopeful, we don't know yet the effect, is the impact of novel technologies. We now can take a human lung and measure and profile every single cell. There's millions and billions of cells. So we have like a Hubble telescope of the human lung. And this should allow us to develop better therapies. So this is our concept of pulmonary fibrosis or an idiopathic pulmonary fibrosis, I will say that I actually don't think that idiopathic is the right adjective now for this disease that we see. Maybe we change the I into interstitial or something, because it's actually, we sort of know what causes pulmonary fibrosis. It is an age-associated genetic interstitial fibrotic lung disease. There's a strong familial background. If you ask people, if you ask them more carefully, there will be a history. Now, many people, especially because IPF is sort of the older age group, when you ask them about their parents or their grandparents, they'll say, well, there was no pulmonary fibrosis in the family. But then think about it, we live so much longer now. So the generation of the people who were born in the 1910s and the 1900s, they died much earlier. So there's a possibility that we never knew that they had pulmonary fibrosis. But apart from that, when we actually do genetic testings or genes, 30 to 50% of the people who don't know that they have familial disease or may have a familial disease have actually genetic variants that have been associated with IPF. The strongest association is basically in what we call the single nuclear polymorphism, basically a random change in your genome, is in the promoter of a molecule called MAC5B, mucin 5B. And this actually, if somebody carries it, it increases their risk to have pulmonary fibrosis in almost sixfold and explains 35% or 40% of the individuals with the disease. So this is a big number. And then there's many other genes that are associated, that are related to what we call telomere function. Telomere is sort of the clock of in our cells in our body, tells us we're aging. And this is found in around 3 to 5%. Then there are cells that are related to the health of the lung lining, again, around 2 to 5%. And then there's genes that are related to our immunity. There's also rare mutations that cause really hereditary pulmonary fibrosis. So pulmonary fibrosis is a genetic interstitial lung disease. Now, this doesn't mean that your family members or your children, your grandchildren, need to live in fear. Because we know that not everybody that carries these molecules develop the disease. This means that they need to keep a healthy lifestyle, avoid smoking, avoid air pollution, but also that they also need to be engaged in research. It's not only about you, it's about everybody around you. So what happens? This is sort of the pathological image of the lung. This is what looks like normal lung, and on the other side is the lesion. And what we think is happening in pulmonary fibrosis is that we have, this is a sort of a cartoon of the lining of the lung, and we have these little recurring micro-injuries that happen all the time to everybody because our lung is in exposure with the air, and the air is never clean as it should be, although we can make it more cleaner. And those of you who know me know that I've been very, very passionate about clean air, and because I think this is probably the best way to prevent lung disease is to have a clean environment. But we have injuries to epithelial cells, then these cells get activated. They call other cells to help them. I'll show it in another cartoon. The cells that we call fibroblasts that make fibers and macrophages, these get activated. They secrete a lot of things that make the lung thicker. And when this happens again and again and again, especially if you have a genetic predisposition, if you have exposure and if your age, you will get pulmonary fibrosis. So this is sort of a cartoon of myself. And I'm sort of saying, as somebody who did this, is, but does this sort of nice film really explain to us what happens? So there's a lot of research, but I will mention one thing because it's important. In 2020, during this terrible pandemic, when everybody was thinking about COVID, we actually, two groups published back-to-back papers in a very prestigious scientific journal, Science Advances. And what we showed is basically that we could profile cells from patients with pulmonary fibrosis. So we actually, you know, our lung is built of many, many cells. They're organized in a specific way. We were able to profiled 500,000 cells from 42 patients with IPF, 38 controls, and other disease. And we're able to identify all the cell types in the fibrotic lung. And we finally have a real atlas. And there's a website, and actually people that, if you rate your iPhone and scan your screen, the QR code, you will get access to the IPF cell atlas. And what happened is that we discovered that in the IPF lung, there's cell types that were never seen before. Profibrotic macrophages. Macrophages is called a macrophage, is big cell that eats stuff. Is a protective cells. And in the IPF lung, there's specific macrophages that seem to drive fibrosis. There's a group of cells in the lung lining that are called aberrant basaloid cells, and there's others. And then there's also other changes that I won't go into it. But now, finally, we have a real catalog, or the first version of the catalog of every single anomaly in the IPF lung.
Papi, settle down.
So how does it look? This is a cartoon of the lung. You can see what we call the airway that brings air. And then there is alveolus, where there is the gas exchange, where oxygen comes in and carbon dioxide leaves. And as you understand, because gas has to pass there, this is really thin and beautiful. There's one macrophage that sort of floats around and senses if there's problems. There's a type 1 epithelial cell, the cell that actually does the work of gas exchange. There's a type 2 cell that is the cell that sort of does the maintenance, secretes things, provides energy, and also this type 2 cell, if a type 1 cell die, the type 2 sort of changes into type 1. So this is the reserve cell. There's a little bit of extracellular matrix. There's an endothelial cell that carries the blood, and there's a fibroblast here and there, again, just to make sure that the lung is structured well. So what happens when there is injury? A type 1 cell dies, a type 2 cell starts to replicating to replace the type 1, right? You need it basically because it serves as the reserve. But if the person is older, if there is a genetic predisposition, actually the type 2 cell probably is not good enough to replace the type 1. And what we get is what's this cause of basically there's a danger signal. And this danger signal is telling the body, oh my God, I'm running out of lung. I need some help. And what the first thing that happens is basically you get activation of the fibroblasts, these cells that try to basically build the scar, close down the wound. You get changes in the phenotype of these fibroblasts to myofibroblasts, so they try to contract, and IPF cell lungs actually contracted. And these macrophages that I mentioned to you become now, again, trying to contain the danger, they become profibrotic. And then a new population of cells, which we call aberrant basaloid cells, also comes in. And we think that they basically also are part of this effort to repair the lung. So this sounds really scary, right? But there's several good things here. The most important thing is that because we now, if we would have looked at the IPF lung and didn't see anything, just let's say dead tissue, then there's nothing to do. And there's diseases that's what we find. But here we see there's a lot of activity. So this suggests that if we could find there's multiple places that we can intervene. One is we could prevent the injury. And this is by healthy lifestyle and avoiding air pollution and smoking and vaping and stuff like this. But also maybe there are drugs that could actually prevent the death of the cells. And then there may be drugs that could prevent the danger signal. And maybe drugs that prevent the activation of fibroblasts or the migration of profibrotic macrophages or the aberrant basaloid cells. So the good thing, the silver lining thing is actually we have a lot to do. And now we actually start to know what we need to do. So instead of just throwing a molecule in the lung and hoping that it sticks, we can actually target the therapies. So for instance, a good example is the drug pamrevlumab, an anti-connective tissue growth factor, targets the activation of fibroblasts. And this is a trial that goes on. And they're recruiting 103 patients. They're going to give an infusion. The phase 3 is open now, and again, people who are interested, you could look at clinicaltrials.gov or other things and look for the trial and see it. So this targets the fibroblasts. The other thing, as I mentioned to you, we found these profibrotic macrophages, and I do not want, the only reason I'm showing you these figures is because they're beautiful and I like them, but also they show this red here is this population of macrophages, these cells that are only in IPF patients or in pulmonary fibrosis patients. And they have specific markers. This is sort of a three-dimensional rendition of every cell in the lung. Every dot here is a lung, and the colors are sort of the markers. So the cool thing is there's actually a fibrosis signatures in these cells. So we should have a drug that, if this drug could wipe out this signature, we could probably affect the disease. And indeed, there is a molecule called pentraxin-2 that has been PRM-151, which is in clinical trials, that seems to eliminate the differentiation into profibrotic macrophages. They did a clinical trial, a phase 2, which showed some promising results. There was actually less decline in FVC, less decline in 6-minute walks, and it was really well tolerated. And there is a phase 3 trial now that's enrolling in multiple places, including Yale. So again, if you could go into the Pulmonary Fibrosis Foundation website, clinicaltrials.gov or others, put in your zip code and actually find if this trial is near you. The other thing I mentioned, and this is sort of is one of our discoveries, is this population of really strange cells, which we call the aberrant basaloid cells. And these cells, there is a lesion in the pulmonary fibrosis lung, the myofibrofocus, and these cells sits there at the edge, and they have interesting features. They're not completely epithelial. They have a bunch of things, but one of the interesting things is that they actually express 3 molecules that make an integrin. An integrin is sort of a surface molecule, and in this case, it's the integrin alpha V beta 6, which is known to activate the key fibrotic cytokine TGF-beta 1. Now, I have to be honest, an emotional relationship with this, because as a postdoc 22 years ago, I was in the group that discovered the role of alpha V beta 6 in pulmonary fibrosis. Actually was a junior contributor to the discovery, and then a bigger contributor to part of the understanding the mechanism. But here's the thing. And now finally, after there's a trial which is in the a trial of testing an inhibitor of this alpha V beta 6 integrin, so targeting the molecule that is expressed in aberrant basaloid cells, a disease-related feature, and that's really exciting that that's starting to recruit. But here's the thing: this trial is based on discoveries from 22 years ago, so completely doesn't make sense. We have to be able to accelerate drug development. Nobody, even, I'm not speaking about patients, but even the people who discovered the drug or the discovery may disappear or pass away before the trial gets to humans or show an efficacy. So we have to go faster. We cannot afford to be so slow. So there is a mechanism which is called drug repurposing, in which you identify new users for approved investigational drugs that are outside the scope of the original medical indication. And the reason this is good is because you build on the experience of others. So this is definitely safer. Usually repurposed drugs have already been found to be sufficiently safe in a preclinical model in humans, and are less likely to fail from a safety point of view, because remember, some of the IPF trials did fail from a safety point of view. So you're likely to give a safer product to the patient. Second, it's faster because a lot of preclinical testing, safety assessment, in some cases formulation has gone away. And the estimate is that it costs you around a third, 30% of the cost to develop a repurposed drug compared to a new drug. And there's multiple examples. I think many of you know about minoxidil, which was developed for hypertension and ended up to be a very effective shampoo for hair loss. There's a bunch of others, and then of course, so it's faster, it's cheaper, and it can lead to new discoveries. One example is actually work that I was, myself and Dr. Antin, Ozerkis, and others have been involved in. This was we were looking for a drug signature that will sort of identify, let's say, a better drug for pulmonary fibrosis based on data that we had. And this was a collaboration between National Jewish, Greg Downey in Denver, Joel Dudley, Christine Becker, and Maria Padilla in Mount Sinai, New York, Faridan Angari, and myself and Dr. Antin Ozerkis at Yale. And basically, we did the screen looking at many, many drugs that have been tested, and we found that this molecule, sarcatinib, seemed to computationally correct fibrosis. And this is sarcatinib. It's orally available molecule that has developed for cancer. It inhibits SRC and ABL. It was most recently tried in Alzheimer's disease in a randomized clinical trial. It had no effect, but it was relatively safe. The drug belongs to AstraZeneca, so we contacted them. We formed a collaboration. We approached the NIH for funding, and we got funding, and basically very quickly did cell culture and animal models showing that sarcatinib reduces fibrosis. This is just example. If you take cells and give them TGF-beta, they become fibroblasts. They become thickened with stress fibers, which we call myofibroblasts. But if you treat them with sarcatinib, they don't do it. The same thing if you give animals bleomycin or TGF-beta, things that cause fibrosis, they develop this image. You can see this dense thing, but if you give them a sarcatinib, they don't develop it. And this seems to be better than, at least in the lab, that the current FDA-approved drugs. But of course, animals and cells don't necessarily are not indicative. But that's what this has led, actually, is into a clinical trial to evaluate sarcatinib in idiopathic pulmonary fibrosis. The trial is run, again, at National Jewish in Denver, at Mount Sinai, New York, and at Yale. We're recruiting. So from the day we thought about it to the day we started the trial, or to the day we were going to start the trial, it took 3.5 years. The only thing is we announced the beginning of the trial on January 13, 2020. You all remember what happened in March, right? So basically we were going to start the trial and boom, it didn't happen. So finally now we're recruiting, and we're way beyond. We actually have spent some of the budget of the trial just because we couldn't really lay off our teams and other things. So we really need the participation of the community. I sort of put on top the emails of the people you can contact in Mount Sinai, New York, in Mount Sinai, New York, and at Yale or at National Jewish. We can also send you the emails, but really we're recruiting and we want this to happen. So this is an example of an effort to repurpose a drug. But we do need a more systematic approach, and myself and others have designed this trial, which is called the Pulmonary Fibrosis Connectome. And the idea is to develop a computational guide to reversing pulmonary fibrosis. The idea that we could identify prioritized list of compounds with a focus on repurposed drugs likely to reverse fibrotic changes in more than one cell type, normalize cellular interaction, affect both the emergence and progression of fibrosis, enhance recovery of regeneration, and hopefully be devoid of adverse effects. This research proposal was funded by Three Lakes Foundation, an amazing group that really is changing the face of pulmonary fibrosis research. The first two projects are run by myself and by Melanie Konigsoff from Pittsburgh, with multiple collaborators and investigators. And basically the idea it would be that we would accelerate finding new drugs for pulmonary fibrosis, and we want to focus on these drugs that have already been in humans and are safe, so we could do the same thing that we did with sorcatinib, and within 2 to 3 years have at least 3 more drugs in trials. So how far are we from curing IPF? The real answer is, of course, I don't know. And I do think that the cool thing is we are actually speaking about cure IPF. If 10 years ago I would say that curing pulmonary fibrosis is possible, I would probably get thrown out of the room. And I think now with the biology, with the understanding that the lung is not just a fibrotic sac of tissue, it's actually an active process of repair, we could probably improve the good repair and eliminate the bad repair. I do think we're really close to have drugs that are potentially even better than the 2 drugs we have now. So I think this is really important, but we really depend on the community. We already have drugs that slow progression. We will have drugs that will stop the disease, and hopefully drugs that reverse the disease, but participation is the key. And the thing for the patient community, it's really important that it's not only that you volunteer for trial, it's that you keep being engaged in your organizations, understand the risk and the benefits, be committed to affecting the power of future of IPF. And again, remember, pulmonary fibrosis is quite, the predisposition is genetic. You're affecting many other people, but be engaged in the community, PF warriors and other things. Be engaged with your physicians. Ask, can I participate in research? And even if the answer is sort of not necessarily, do the searches and see. And hopefully the other thing is, just by participating, the notion that pulmonary fibrosis is sort of this rare disease that nobody's heard of will change, and this will affect also lawmakers and others. So I want to thank you all for participation. This is sort of a photo of the Yale LD Center of Excellence, not including the lab people, and so probably a third of the participants. If you need to contact us, ildinfo@yale.edu is the email. We have an amazing group of physicians, nurses, respiratory therapists, support teams, and of course, research staff and PhDs. You can follow on Twitter or Facebook, both myself and Yale PCCSM. And you can always click on the hashtag CureIPF and you'll find some updates. So I really thank you all for coming in today. Seeing your faces on my screen is really an inspiration and a reminder to overcome the grinds of daily difficulties and push, because we want to cure people and make their life better. And of course, thank you, Bill and Shelley and the PF Warriors. And we're available to answer any question you have.
Dr. Kaminsky, when I tell you, you knocked it out of the park, you knocked it out of the park. Dr. Antin Ozerkis, what a lucid, clear description of this terrible disease we live with. Both of you, thank you so much. We're going to open up for questions from everybody. I know we have a number. Shelley, you have already gone through a qualification, I think, and sent a number of questions to Dr. Kaminsky and Dr. Antin Ozerkis. Do you want to lead the questions and direct them to them for answers?
Yes, we have, oops, oh my goodness, I'm sorry about that. We have some questions that have been submitted. I'm going to start with probably the one that I think is most interesting and important. Will the cure for PF heal the scarring, or will it just stop any further scarring for the patients who have it? Dr. Kaminsky?
So I think that the main thing that has changed in the last 15 years is that we understand that fibrosis is a dynamic process, right? So there's a mix, even in when we take an IPF lung from a patient who has undergone transplant, these lungs really look bad, right? As bad as we can. But even in these lungs, we see healthy cells and we see normal regions. And I think there's a mix of activities. So the way I look at it, we first slowed down fibrosis. That's done now. Now we want to stop fibrosis. And then we want to reverse fibrosis. And I think if we stop it, you'll start seeing some reversal in the areas of where we stopped it, because the fibrosis sort of have a pattern of progressing in the lung. One thing I want to mention is something that I would not have believed. So there's a disease that shares a name with idiopathic pulmonary fibrosis, which people know much more about it, completely unrelated, which is cystic fibrosis. It's a genetic disease, affects young people. We know the mutations. When I started training, we expected them to live to teenage years or 20 years. Through improvement in care and nutrition and infection management, they increase their life expectation in people to around 40 or 50. But they never got better. It was sort of slow decline, and they needed lung transplant. In the last 2 or 3 years, and the director of our cystic fibrosis program every now and then shows me the data. We have drugs that actually improve the pulmonary function of these patients. So somebody who was going always downhill then was happy to be stable. Now is getting better. And I think that's what we should aim. And the ambition in that disease of actually reversing the disease is actually the ambition we have to put. If we don't say we're going to try to reverse it, we'll never be able to do anything, right? If our aim is only to slow down, maybe we fail even at that. So we have to be ambitious. I can say everything we see in animal work suggests that you could reverse fibrosis. And I don't think that's unique to the animal. It's just that, we never can give the same doses or the same location to humans. We haven't tested all the drugs. But yeah, I think we want to slow down. Now our aim is to stop, and the next aim is to reverse.
May I ask what you're doing with the animals that appears to reverse the fibrosis?
So it's important to understand that in animals, what we're studying is fibrosis that take a few weeks to make. So a lot of things will reverse it. It's like you can almost think about it as a small scar you had on your hand, right? Sometimes you get some surgery and it looks like there's going to be an ugly scar, but a couple of years later, you don't even see anything, right? Mm-hmm. So I think some of these scars resolve spontaneously. So I think we, both by therapies that affect the health of the epithelial lining of the lung, we can reverse fibrosis. Some therapies that affect the macrophages can reverse fibrosis in animals. I think but these are therapies these are interventions that to have efficient therapies, probably a few years to get into humans, the same thing.
Okay. Also, we had one about when you're doing trials, do you get a lot of anecdotal data, and what do you do with that, and how do you identify it?
I'll tackle that one. So part of the reason that there is randomization, as I had mentioned, is because there always is going to be a lot of information that comes in, and you never really know what is relevant and what is not in any individual person. And so when a participant is in a trial, we're collecting every little piece of information, from do they have a headache one day to did they get hospitalized? Because if it turns out that 2 times the number of people in the treatment arm developed headaches, well, maybe actually that is actually a side effect of the drugs. But if it balances between the groups, it may just be that people sometimes get a headache. And so we really try to gather all that information and make sense of it. And that's really the purpose of running it in this way, because sometimes it's more serious than a headache, and you wouldn't know it unless you studied it in organized fashion.
Okay. We also have some very good questions coming in on the chat. If Dr. Kaminsky, are you looking at any of those chat questions? There's a very good one from Talina Koch about the Panther study that was mentioned in one of your slides. It says, while we know the study showed the cocktail was causing too much high mortality, at some point it was also found, as I hear, that prednisone is contraindicated for IPF specifically. Is that really the case? I've been trying to find a paper on prednisone itself for IPF, but all I find is the Panther study cocktail. Does prednisone cause IPF to progress?
I can also take that question. So prednisone alone has never been studied in IPF. This is, if you're looking for a specific paper on IPF treatment, you will not specifically find prednisone as a treatment. I want to be clear, and I tried to mention this as I was discussing the Panther paper, but I want to make sure that people understand that there are different reasons for treating with different medications at different points or in different diagnoses. So, for example, what the Panther study showed was that for IPF, so specifically IPF, for patients with moderate disease, that we should not be using this cocktail of medications. What it did not address was other forms of pulmonary fibrosis. So for example, somebody with rheumatoid arthritis, and that's why they have their lung disease, it might actually be that prednisone and azathioprine would be good medications in that situation, because sometimes there's inflammation. Also, IPF can sometimes have what's called an exacerbation, where somebody might be living their life and doing okay, maybe slowly getting a little bit worse, and then suddenly something happens where things get a lot worse and there's new inflammation in the lung. And with an exacerbation, sometimes it's triggered by something, sometimes it's triggered by an infection, sometimes we don't know why it's been triggered. But in those situations, we often do use prednisone when there is new exacerbation. There have not been a lot of really great studies in that situation. And so this is based on our medical knowledge and experience, but not based on trials. But people are trying to study exacerbations now. And so we may have more information about how best to treat an exacerbation in the future. So I hope that answers that question for you.
Okay. Another one that came in beforehand, it was thank you for taking care of us and your dedication to finding a cure. I have pleuroparenchymal fibroelastatosis. Will these therapies help those of us with PPFE as well, or is the structure of our lung disease just too different? I hope I pronounced that okay. Dr. Kaminsky?
I actually, this is a really important question because we actually don't know enough about parenchymal fibroelastasis, pleuroparenchymal fibroelastasis. And for instance, my lab doesn't have a single sample right? We're just now getting some. So, and believe me, my lab has the biggest collection of lungs probably in the world. So I think it's actually really important to push for specific research. I have a sneaking suspicion that some of the mechanisms are going to be very similar. I actually don't think that, let's say, some of the cells we found are like the aberrant basilar cells are probably not there. So it's only the beginning. And I actually think there should be a little bit of a push for NIH to encourage research, and also with the patient organization and the foundations to help us to get the biological information. I do think that everything that will affect fibrosis in a big way will have some effect. That's what we learned from the clinical trials that Dr. Antin Ozerkis presented, is that what worked in IPF usually works in almost any fibrosis that progresses. So I think there's a cause for hope, but we definitely need more data and more information.
Okay, another question: with the rise in pulmonary fibrosis diagnosis, RA among younger minorities, how effective, or your outlook, are clinical trial outcomes? How is the population being educated about RA, PF, and the treatments?
So I think this is a really important question, because a lot of the focus in the pulmonary fibrosis world has been initially on IPF itself. And I think that IPF obviously is an incredibly important disease to focus on, but we're now really expanding that focus to other forms of interstitial lung disease. And the kind of lung disease you get from rheumatoid arthritis is absolutely one of them. And there are more and more studies aimed at that. We did not discuss that specifically during this talk, but it's an incredibly important piece of research, because people with RA-ILD have serious lung disease, and absolutely it's in need of study. Those people were included in the studies I presented where nintedanib can slow progression in progressive fibrosis. Those studies did allow patients with rheumatoid arthritis lung disease in. And I think that that is not only going to be the future in some trials, but hopefully there will be focus on those diseases as well, because there may be differences in those kinds of lung disease as well. But I think it's very important that we focus on all types.
Oh, thank you. And this question: I was in the Galapagos Isabella trial for 2 years and 4 months, which was ended in March this year due to safety issues. I guess that means COVID safety. I don't know. Do you know what the safety issue was and how serious it was?
I can answer that if you'd like, or
yes, please.
So yeah, so we only know what the company released which was that there was a safety issue. And when we say safety, it was not a COVID related safety issue. So as I was mentioning, that there's always people overseeing the trials in the background. There's something called the Data Safety Monitoring Board. And so when a trial is running, they will analyze the data periodically through the trial to see, is there any sign that the people receiving the drug are doing worse in some way, or is there some safety issue that's coming up where there's an event that's happening more often in the people getting the drug as compared with the placebo. I told you that the people investigating the trial don't know who's getting what, but there is a safety group in the background monitoring this. And so that's what happened with the Galapagos trial. They looked in the background, they saw that there was a difference, and they stopped the trial because they thought it was not safe to continue. I can't give you more than what the company has released at this point, but they will eventually give more information to the public.
Okay.
Yeah, I want to highlight, this is an important point, is when we run a clinical trial, and we do, sometimes your patient walks in their garden and and twist their ankle, and you start thinking, is that the drug, right? And you don't know. It's really scary, and it's your responsibility, right? It's really our patients entrust their lives with us, right? So this is not easy, and people tend to think that some people think about drug companies as these bad organizations, but the people who work in them are committed to the health of the patients, and it's physicians and you go into really difficult discussions sometimes because you don't want to kill something that's good, right? A drug that could work because you misinterpreted the results. But most companies will do. It's a little bit like with the COVID vaccine when they stop them for a few weeks. Most companies will stop, will look at it. All these groups are independent, and there's we've had several drugs in the last few years that were stopped for a little bit of safety concerns. And it's really important to know that this is happening and monitored very closely.
Yes, and I understand that. And I'm sorry, I'm actually the one that put that question out. And there was the only information I've been able to get out of the investigator is that possible death, which was not what I wanted to hear. And so my real question is there a standard of time after which they have to release? Because I've been in 3 trials. This is my 3rd trial. I'm getting ready to go on a 4th one. And the issue that I had with the first 2, not the 3rd so far, is that after a year, I was not able to get any information about effectiveness or whatever. It did come out 18 months later that it just didn't make sense to go ahead with it, but there weren't safety issues. So is there, who determines what the time limit is on giving that information out as to after investing 2 years and 4 months in it?
So here's the thing. So it depends on the stage of the trial. So remember, we look by the time the drug gets into humans, it's already been in mice, rats, usually non-human primates, so monkeys and dogs. So you already have a good sense of safety. And then you do a phase 1 in healthy people, which, but the number is small, right? And you do a phase 1 in sick people, and then you do a phase 2. All of this tells you that the drug is generally safe, right? It's not a huge risk. But the problem is that when you do it in phase 3, is you're looking for the rarer side effects, right? And these are also harder to adjudicate or understand because, in every study that you do, also people that don't get the drug die or have side effects. So it's really hard, and sometimes the process is really long. What will happen usually is that the safety and monitoring board will dictate the decisions, whether to stop the drug, how do you look at it, how do you assess causality, is it related to the drug or not? And sometimes the problem that the trial may be stopped, but the announcement could take a year later just because it's really hard to actually understand. I think the good thing now is, when you get to a clinical trial, they draw your blood, right? And they do all these tests. And many of the things that we do in the testing of a trial actually have to do more with safety than with anything else. And that we're driven by some of the information from the animals that will tell you, okay, if liver function tests are going a little bit up, it's okay to continue. But if they get a little more worse, we have to stop.
There is, Professor Kaminsky, there is a rule now, a federal rule for clinicaltrials.gov that companies sign up to a year after the primary analysis. The
Yes.
data should be submitted to clinicaltrials.gov, both for efficacy and safety. And that is something that there are organizations monitoring how that is being done. Not everyone is complying with the 1 year. So I think the industry has definitely got a ways to go to be fully compliant. There is an agreement that some phase 1 trials can delay sharing their data. But the rule is a federal rule now, and it's very explicit.
Yeah.
Thank you. Can I say one thing? I think it was a phase 3, so that at the end of 3 years, I would be finished. But then the way it was set up was that after everybody that was in it finished, that I would be able to, I'll stay on the drug and I'd be able to keep going on it. So it was a phase 3. So 1 year I can understand. It was 18 months on the previous 2, but I don't know when that law went into effect. So the other thing I want to say is I am definitely going into my 4th one now here in a month. I definitely don't want to give anybody any negative vibes about clinical trials. So understand that. I have been diagnosed for 8.5 years. So like I say, I will try anything, including all the health things that I did, to stay alive as long as I am. My hero has been Bill Vick from the old Discovery Channel thing that I saw years ago, right after I was diagnosed. So I will do anything, and I want everybody to get involved in clinical trials a lot, and I will never give up, quite frankly. Second thing, there used to be, I am getting in, I ran up against the problem of a 5-year limit after diagnosis for most of the major trials. When I first, and then Galapagos came down, and I got in under 1 month under the 5 or 6 year limit, whatever it was. So like I say, I got in on that and thought that I was told I'd never be able to get in one again. I am getting ready to go into some, but I like everybody to kind of understand what is, it seems to be up to each individual drug company as to how they're going to do it. And I got to tell you, there are people living past 5 years now with IPF.
Thank you, Fred. Yes, that's really important. And actually, that brings us up to another.
I want to say something. I want to say something
Okay.
about this. And this is so important because there's 2 things that are important here. One is, and that's going to be a little bit sort of my other side. I actually believe that all clinical trials have to be publicly available without identifying their patients within a year of finish. So not only the results, the raw data should be out there for other investigators, but it could be that the original group didn't find something. Maybe the drug actually works in a subgroup. So that's one thing. So I actually think, especially nowadays with computations and big data and other things, this has to go. The reasons companies don't do it is one is inertia. It's not only that they're afraid, it's just every time you share information, you need to work. The other is this perspective, the limitations, like so many drug trials now used to have an age limit, an upper age limit on them. I don't agree with that. There's no real reason for this. The same thing, the 5 years, there is a real reason to make sure that your design is balanced. So you're not studying one group of patients or the other, that you know what you're studying. But many of these limitations are historical, because when we started studying IPF, we wanted to have the sort of the cleanest population possible. And also we didn't know if we could recruit patients, but here's the reason I'm interfering is this is where the voice of patients and their families is so important. You know, if somebody is willing to write an editorial and post it on Medium, we could give it to NIH, we could give it to FDA. It's not, these things are not unique to IPF, but it's really important for patients, families, friends to have a voice.
Yeah. Dr. Kaminsky, I know you have an appointment you have to go to. We're going to close down after this short announcement, but let me say, as a patient, I participated in trials I think are incredibly important. Without trials, we will never see a cure. If you want to be part of the solution and not part of the problem, I would encourage you to investigate them. There's a number of resources. I'll be sending links out to two. One's on the PFF. They've done a good job on it. There's another organization that also has kind of a front end for clinical trials. And we're going to help you understand where the trials may be in your area. And as Fred says, and Fred, you're going on number 4 now, because of that, we've made advancements. You are a participant in helping find a cure for the rest of us. I think we all owe it to ourselves and each other to help, find a way to help and find a way to give. And kind of closing down, I just have to say, Dr. Kaminsky, Dr. Antin Ozerkis, what a great presentation. It was a learning experience, and to me, a motivational, inspirational experience. And I want to thank both of you and Yale Medicine for all you do for all of us. And you touched on something that's dear to my heart, is that we, as in the patient community, our caregivers, our families, and us, we have to find one voice and one message and knock on the door of NIH and raise awareness of this disease and what it really means to us, so they understand the urgency of finding a cure. Because without a cure, we know the outcomes, and I want the outcomes to change. So again, thank you so much. And we'll be putting this online. You'll be getting an email probably on Monday from us with some links. The video itself, you'll have access to it. It's a video you can share with others, and we encourage you to do that. We had a few hiccups here, like I was invisible at the first part of the meeting, but I took off my cloak of invisibility, so that worked out okay. And I want to thank you all for being here. Love my warriors, love all you guys, and thank you so much.
Thank you so much for inviting us. It was really a pleasure to be with you all.
Yes, thank you all.
Thank you. Thank you. Bye-bye.
Bye-bye.